If you were to feel your pulse right now, you would probably sense your heart beating in a very regular and coordinated rhythm. But have you ever considered what may happen if that rhythm wasn't so regular? Irregular heart rhythms are referred to as cardiac arrhythmias. During an arrhythmia, your heart may beat too fast, too slow, or in a very irregular fashion. This can be dangerous and even deadly, as the primary function of your heart is to serve as a pump, pushing blood throughout your body. However, during an arrhythmia, this pump function can become compromised. Therefore, the goal of my research is to determine why and how arrhythmias occur, but in a unique patient population. Many people in this room have probably heard of epilepsy, which is a neurological disorder that causes seizures. I work with a mouse model of an epilepsy disorder known as Dravet syndrome, which affects children. Individuals with Dravet syndrome have an incredibly high risk of developing a fatal complication of epilepsy disorders, known as sudden unexpected death in epilepsy, or SUDEP for short. No one fully knows why SUDEP occurs, but researchers believe that cardiac arrhythmias may majorly contribute to SUDEP. But to determine what mechanisms in the heart exactly may lead to arrhythmias in SUDEP and Dravet syndrome, we have to first consider the pathology of this disease. Dravet syndrome is typically caused by mutations in proteins known as ion channels that sit in the perimeters of our cells. These ion channels serve as almost little ion bodyguards, only allowing a certain amount of ions into our cells at specific times. However, in Dravet syndrome, when these proteins are mutated, this bodyguard function can become lost. This can significantly impact the environment of our cells and also the function of other cellular structures or organelles, such as mitochondria. You may have heard of mitochondria before, being referred to as the powerhouse of the cell. This nickname came from mitochondria's ability to essentially act like little powerhouses in our body, generating tons of energy in the form of ATP to support metabolism. However, as a consequence of mitochondrial ATP production, they also produce chemicals known as reactive oxygen species, or ROS for short. When ROS accumulate to high levels within the cells of our hearts, this can cause cellular dysfunction and also these cells to die, which can lead to arrhythmia. Therefore, my thesis is that compromised mitochondrial function, whether that be ATP or ROS generation, can promote arrhythmias in SUDEP and Dravet syndrome. My hope is that the results of this work could potentially promote the formation of new mitochondrial targeted drugs to prevent arrhythmias and sudden death in patients with epilepsy. Thank you.